So hello everyone and welcome to this EBI webinar. My name is David Armstrong and I'm from Protein Data Bank in Europe. And I also have Matthew Conroy here with me who will be answering any other questions you have via the chat box. Today I'm going to be giving you an overview of some of the updates at Protein Data Bank in Europe. You can see there's some contact details there if you want to get in contact with us. I'll put these up at the end as well so that you have these. So a quick outline of what's going to be in this webinar. I'll start off with a very brief introduction to who we are at the Protein Data Bank in Europe. I'll then move on to talking about some of the recent search updates. These include some new advanced search features, sequence searching, which we have through pHammer. There's some more features on the search page, and again, I'll give you an overview of these. And finally, just some of the other recent updates we've had at PDBe. This includes the ability to more easily find RNA molecules at PDBe, and also for highlighting cofactors. So a brief introduction to us to begin with. So first of all, the Protein Data Bank, the PDBe. So this is an archive of experimentally determined structures. What this means is that we have to accept data only from a certain set of techniques, that's crystallography, electron microscopy, and NMR, nuclear magnetic resonance. And that means that we can validate the data against the experimental data that we also accept as well. The other important thing to say is that it's biological macromolecules. It isn't just proteins. We also have structures of nucleic acids and complexes of both proteins and nucleic acids. So we are part of a wider worldwide protein data bank, and this also includes our partners in RCSB PDBe in the US and PDBJ in Japan, and also BMRB, who deals specifically with NMR data. And you can see from this map that there are different geographical locations highlighted for different sites. So we accept all depositions here at PDBe from Europe and Africa, RCSB from the Americas, and PDBJ from Asia. However, we all do this in the same way, and all of this data is put into a single FTP archive, one single archive that you can get the data from. So this ensures that it's consistent wherever you get the data from. So the next step is from that archive, we then serve you the data on our individual websites. And because we have separate websites, we can do this in slightly different ways and provide you with different tools in order to do this. And that's where being at the EBI comes in very handy, because we are in the middle of a whole load of other resources. You can see us here in the molecular and cellular structures cluster, but we have a whole host of other databases, and we can use that additional biological information to give much more context about the structures that we have. So to begin with, I'm going to give you an update on what we've done with the search here at PDBe in recent months. We've added much more advanced functionality to our search at PDBe. So hopefully this will help you find the structures that you are looking for. We also wanted to simplify how you could access these advanced search features. We only have one search box still on our homepage. You can see this here. And the autocomplete search that you get through this search box will automatically integrate into our advanced search. So essentially it is all one search, but we've added a lot more features to it. There is the opportunity to start from scratch and go directly to the advanced search form. And if you click this link here, that's what will happen, and you'll be taken through to this page here with the advanced search form. You have an option then to select the search fields that you want to search for. So you can either use the drop down or start typing, and the autocomplete will get a selection of different fields. So for instance, if I started typing molecule, you can see here that there's a list of possibilities for molecule as search fields. So you could search by molecule name, molecule type, or expression method, for instance. Once you've then chosen your search field, in addition to that, we also have a lot more electron microscopy data in our search as well. So some examples here of EM resolution, specifically for electron microscopy entries, reconstruction method, and a lot more electron microscopy categories. So once you've decided which field you're going to search from, so for instance, in this example here, I'm searching in the experimental method. If you start typing, you'll get an autocomplete list of possible choices that you can have for that field. So here I've started typing diffraction, and you can see I get a list of different techniques, different experimental methods that I can sort by. And the number next to it gives the number of instances of that search term in the whole archive, and that includes unreleased and obsolete entries as well. We've also added ranges to our search form so that for numerical values, you can have much more control over the values that you're searching for. So in this example, I'm looking at resolution, and I'm able to specify a particular resolution range to search by. Once you start adding additional search fields, you then have various different ways to customize your search. So this includes the option of adding conditions such as and, or not. And you can also specify whether the term that you search with is contained within that field or whether it's specifically equal to. So that can give you a bit more flexibility on how you search. If you do change your mind about a particular search term and you want to remove it from the form, you can use these crosses in the corner of the search terms, and those will be removed easily from your search form, and you can add new fields if you wish. So once you submit your search form, you will then be brought back a list of results, and you will also get at the top left here, it will show you what you have searched for and return those back to you as an option. And again, these have crosses on so that you can remove these if you change your mind. You can also still select facets as before. You can see those down the left-hand side, and these will also be integrated within our advanced search form. So for instance, if I decided to search by assembly polymer count, I might want to narrow my search down to only look at the monomers in the search results. And if I click on this, it will take this term and add it to my advanced search form, as you can see in the top left here. So all of our different search functionalities are all integrated into one handy form. Okay, so before I go on, we'll just have a very quick poll to just get some information from you, and yeah, if you could just answer that, we'll be back in 30 seconds or so. Okay, thank you very much for voting in the poll. So it seems that most of you search for molecule information, and it seems to be the way that you most want to find PDB data, which is great. Okay, so I'm going to now move on to the next part of the webinar. We have integrated pHammer so that you can now search the PDB by protein sequence. We have integrated this into our advanced search form. If you choose the search field of pHammer sequence search, you are then able to input a protein sequence, so you can just paste your protein sequence straight into the box. This is not currently available for nucleic acid sequences, but you can search for proteins this way. If you then click submit, this will send your sequence through the pHammer server, and we get our results directly from pHammer. And what they give you is a unique result for every unique protein sequence in the PDB. At the moment, this is only available through the Entries tab, not through our other tabs at the top here. The output that you get includes a sequence alignment. So you can see in this example, the alignment between the query that you searched for and also the target that it has found in this entry. And again, you can see how good the match is between the two. There are also various statistics to give you an idea of how good the scores are. For instance, there's an identity percentage, there's an e-value, and we actually order our search results by e-value. And you also get a chain ID at the bottom left there, so that you know which specific chain in the entry has been matched by the sequence search. Finally, as pHammer returns only identical sequences, you can then click this button here to check for all the other entries that have an identical sequence so that you can compare those structures. There are a number of features that have been added to the PDB search to help you easily analyze the results of your query. So one of these is adding visualization directly to our search results. So on each of our search results on the entry page, there is a 3D visualization button, and this will open our Lightmole viewer directly from your search results. So this has all the features that we would have on an entry page. So if I was to click on a particular residue, it will zoom in to that particular place. And for crystallography entries or for electron microscopy entries, it shows you the map, so the real data behind the structure, and you can see how well that fits. Some of the other features include the ability to apply the validation report to your structure. And if you were to hover over a particular residue on the validation, at the top left, it shows you what the actual validation issues are. Another thing we've also added is the ability to color by domain annotation. And all of this, remember, is on our search result pages, so you can quite quickly see where is the domain I'm interested in my search results. We've also added the ability to directly download files from your search results. If you, for instance, wanted to download the entry files, you can click on this download files button here, and you'll get various options of files to download. So if you were interested in the entry file, you can get hold of the archive files here. So the MMSIF is the archive file. Or if you were wanting to get hold of a PDBe file instead, you can also do that here. We also have additional files for download in these other sections. For instance, you can get hold of the EDS map for crystallography entries. You can also get assembly files so that you can download the most likely assembly as a structure to look at in a separate viewer outside of our website. There's also files to do with validation information and SIFs mapping to UniProt as well. We also allow you to download your search results as data files so that you can analyze them afterwards. So if you're not interested in the nice graphical representation we give you and you want something that you can use in a more analytic way, you can do this. So you can either select all the entries on the page or select individual PDBe ID codes. If you don't select anything, then it will automatically default to the whole search. By clicking the download button in the top right-hand corner, you'll then be brought up with a form which gives you the list of different fields that you can download information about. So if you select all the ones you're interested in from this list, you can then choose which format you would most likely to get the data in. And finally, click download, and this will enable you to download this information to your desktop to then be analyzed on a different surface if you wish. So we'll have another quick poll now, and we'll give you a minute on that, and I will come back and continue from there. Okay, thank you for voting in that poll. It seems that most of you are interested in looking at results in terms of PDBe entry or individual macromolecule. However, there are a few of you who are interested in looking at complexes, which is very interesting. Okay, so I'll now move on to talking about some other updates we have here at PDBe that have come in the recent months. We have made RNA molecules much easier to find at PDBe, thanks to mapping these molecules to Rfam data. So Rfam are one of our sister databases here at EBI, and they classify RNA into families based on their sequence similarity. So once you have these sequence domains classified, you can then map these onto the RNA molecules in the PDBe, and this enables you to much more easily find RNA molecules in the PDBe. What we can also do is include these domains in our web components. So for instance, in Lightmole, we can highlight where particular Rfam domains are within a structure, so you can more easily see where they're positioned. So if you did want to search for RNAs at PDBe, you can use our standard search bar on our homepage or on any of our web pages, pdbe.org. So for instance, if I was looking for tRNA, I could type tRNA into the search bar and I'll get a list of autocompletes. That will include sequence family. So we have a whole list of sequence families, which includes protein sequence families and now Rfam data as well. So if you look a bit closer there, you can see we have 2 results back for tRNA because we also get clan data, which is the top one, which is a higher level information from Rfam. And we also get the specific Rfam family, which in this case is tRNA. If you want to search in slightly more detail for Rfam IDs, then again, you can use the Rfam, the advanced search form. If you look for Rfam accession in our search fields and type in tRNA in this case, you can see that we're just getting back an Rfam domain because this is specifically looking for individual Rfam domains. And this filters out the noise from the other sequence domains as well, so you can get specifically what you're looking for. We've also changed the way that we present the results to you. When you click on our macromolecules tab, no longer is it only proteins that you're given back. You also get RNA and DNA molecules shown as individual molecules. So in this case, we show the best example from all the tRNA FBETs that we have in our search. Finding structures of enzymes with bound cofactors has previously been quite difficult in the PDBe, so we've aimed to try and improve this. So organic enzyme cofactors are very highly involved in enzyme reactions, but actually finding them in the PDBe has been quite difficult. So we've been collaborating with the Thornton Group and CofactorDB, who are also based here at EBI. And with their help, we've helped classify likely cofactors within all the protein structures in the PDBe. So now you can more easily find these by searching at PDBe, and we highlight these more prominently in entries now. So if you did want to search for particular cofactors, you can find this in our advanced search form. So if you choose compound cofactor class as a field, and you can get a drop-down list here of all the cofactors in this class. And if you choose one of those specifically, you can then search the whole PDBe for that particular compound cofactor class. We also display this information on our entry pages. So in the ligands and environment sections of an entry page, if one of the ligands is classified as a likely cofactor, then we label this here in the ligands environment page. So for this entry, for instance, TDP in entry 5HHT has been classified as a likely cofactor. If you were to click on that ligand and go through to the specific ligand page as well, again, we highlight this information and let you know that it's got a likely function as a cofactor. And there's also various other links out on the right-hand side if you do want more information about this. Okay, so that's it for the webinar today, and thank you very much for your attention. I would like to ask if you do have any questions, please add them in the comments box, and we'll try and get back and answer as many of those as possible before we finish. We also have some contact information there if you want to follow us on social media or get in touch with us in other ways. Thank you.